Hi everyone. Today we will discuss the RNA synthesis or the transcription process. Here we will discuss what is transcription, the unit of transcription, and the transcription in both prokaryote and eukaryotic organisms. Transcription is the process by which the information in the strand of DNA is copied into new molecule of mRNA or messenger RNA. This is to ensure that the DNA in the nuclei of the cells safely and stably stores the genetic material. Transcription process is being carried out by RNA polymerase. RNA polymerase is a multi-unit enzyme that synthesizes RNA molecules from a template of DNA. This is different with the DNA polymerase, which synthesizes another DNA strand from that of a DNA strand. RNA precursors include adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, and uracil triphosphate. You have to remember here that thymine in the DNA is being replaced by uracil in the RNA molecule. And just like in the DNA replication process, transcription moves from a 5' to a 3' direction. In this figure, remember that despite the fact that genes are present in both strands of DNA, only one of the two strands of DNA is transcribed by RNA polymerase for any gene. The E. coli or Escherichia coli RNA polymerase, RNAP, is a multi-subunit enzyme composed of 5 subunits, including 2 alpha subunits, a beta, beta prime, and an omega subunit. These 5 units form the RNA polymerase, core enzyme responsible for RNA synthesis using DNA as template and ribonucleotide as substrate. In the process, RNA polymerase binds with the sigma factor. The sigma factor recognizes promoter and initiation site. A promoter is a region of DNA upstream of a gene where relevant proteins such as RNA polymerase and transcription factors bind to initiate transcription of that gene, leading to RNA synthesis. Once promoter and initiation site is recognized, transcription begins, sigma factor is released, and RNA elongation follows. Transcription will only stop when the termination site is reached, after which the polymerase and the RNA is released. Remember that the promoter is a site of initiation of transcription which is recognized by the sigma factor of RNA polymerase, and that transcription stops at specific sites called transcription terminators. Unlike DNA replication, transcription only involves a segment of DNA for the synthesis of an RNA strand. The figure here depicts the interaction of RNA polymerase with a bacterial promoter. Remember that the sigma factor recognizes 2 highly conserved regions of the promoter. The negative 35 region, which is the Tata-GACA found 35 nucleotide base upstream of the gene, and the Pribnow box, which is located at 10 nucleotide base upstream of the gene. Only when the sigma factor recognizes it that transcription starts again, sigma factor is released, and elongation proceeds. From transcription to terminate, this is governed by specific DNA sequence. Just like what we said a while ago, transcription terminators should be recognized. We have 2 types of transcription terminators. We have intrinsic terminators, which terminates transcription without additional factor, while the row-dependent factor termination requires row protein to recognize specific DNA sequences for termination to proceed. The unit of transcription is a sequence of nucleotides in DNA that codes for a single RNA molecule. This is a DNA strand bounded by sites of transcription of DNA to RNA and is initiated and terminated. The strands are later on translated into proteins, but some RNA are not being translated. Say, for example, rRNA and tRNA. rRNA, or the ribosomal ribonucleic acid, have three types, which includes 16S, 23S, and 5S. tRNA, or the transfer RNA, on the other hand, carries the anticodon needed for protein synthesis. These two, the rRNA and the tRNA, are needed for translation to proceed. Finally, mRNA is the segment of RNA that carries the code for protein synthesis. This figure, on the other hand, shows the operon and polycistronic mRNA structure in prokaryotes. In prokaryotes, in addition to not having introns or non-coding segments, they often have genes clustered together, which are transcribed all at once in a single mRNA. An mRNA encoding a group of co-transcribed genes is called polycistronic mRNA. In the figure, the polycistronic mRNA actually contains 3 open reading frames, or ORFs. On the other hand, operon is defined as a group of related genes co-transcribed on a polycistronic mRNA and allow for the expression of multiple genes to be coordinated. Specifically, when we refer to the genes, we call it operon, but when we refer to the mRNA, we describe it as a polycistronic mRNA. In transcription, archaea contains only a single RNA polymerase, which resembles a eukaryotic polymerase II. This is very different with bacterial polymerase, which is a multi-subunit enzyme composed of 5 subunits, including 2 alpha subunits, a beta, beta prime, and an omega subunit. These 5 subunits form the RNA polymerase core enzyme responsible for RNA synthesis using DNA as a template and ribonucleotide as a substrate. The similarity in RNA polymerase between archaea and eukarya supports their close phylogenetic relationship. Archaea have a simplified version of eukaryotic transcription apparatus with promoters and RNA polymerase being similar to that of the eukaryotes. However, their regulation of transcription have major similarities with that of bacteria. On the other hand, remember that eukaryotic genes have exons and introns, which are rarely found in archaea and are not only found in their rRNA and tRNA genes. Also, archaeal introns are excised by special endonucleases. For eukaryotic RNA to be translated, pre-mRNA needs to be processed prior to translation. Specifically, spliceosome is assembled to cut the intron, thus forming a lariat, and bind together the exons, which will then be translated to proteins. The spliceosome is a large RNA protein complex that catalyzes the removal of introns from nuclear pre-mRNA. A wide range of biochemical and genetical studies shows that spliceosome comprises 3 major RNA protein subunits, including U1, U2, and either U4 or U6. Aside from that, we define lariat as a lasso-shaped structure formed from the fragment of RNA to be excised during the process of splicing out the introns in the primary transcript of eukaryotic genes. Finally, remember that RNA splicing performed by spliceosomes take place in the nucleus, removes introns from the RNA transcripts, and binds together exons which will be then translated into proteins. Finally, RNA capping and poly-A tail are observed in eukaryotic RNA processing. RNA capping is the addition of methylated guanine to 5' end of the mRNA, while poly-A tail is the addition of 100 to 200 adenylated residues in the 3' end of the mRNA. What is the significance, therefore, of having RNA capping in the 5' and the poly-A tail in the 3' end of the mRNA? Well, both makes the RNA molecule more stable, prevents its degradation, and allows the mature mRNA to be exported from the nucleus to the cytoplasm for it to be translated into proteins. We will end our discussion here. Next time, we will discuss protein synthesis. Thank you and see you next time.